Today, I'm going to talk about target repositioning, a novel method to predict therapeutic targets for various diseases. Therapeutic targets are biomolecules leading to therapeutic effects for disease treatment. Identifying therapeutic targets is important in drug discovery. However, therapeutic targets that can be identified using pathological knowledge have been thoroughly investigated. So, developing computational methods to predict therapeutic targets is needed. We propose a novel concept of target repositioning to predict new therapeutic target indications for a wide range of diseases. For the prediction of therapeutic drugs, drug repositioning is popular. Drug repositioning identifies new indications for known drugs. We propose the extension of the concept of drug repositioning to target repositioning. The bottom figure implies that target proteins have the potential targetability for different diseases than the original diseases. Target repositioning predicts new indications of known therapeutic targets. The purpose of our study is to develop a computational method for target repositioning. We try to predict novel therapeutic targets for various diseases. We propose two methods by using disease-specific transcriptome data and target gene-perturbed transcriptome data. First, we constructed disease-specific gene expression signatures from gene expression profiles of patients. Next, we constructed two types of gene-perturbed transcriptome signatures of target proteins. Target gene-perturbed transcriptome signatures represent gene expression patterns following gene knockdown or gene overexpression of target genes. For inhibitory target prediction, we use target gene knockdown signatures and disease-specific gene expression signatures. For activatory target prediction, we use target gene overexpression signatures and disease-specific gene expression signatures. Next, I will show our proposed methods. The first one is inverse signature method. We focus on the inverse correlations between target signatures and disease signatures to predict therapeutic targets. We predict target proteins with highly inverse correlations as candidates for therapeutic targets. The next one is trans-disease method. In the trans-disease method, we predicted therapeutic targets by multitask learning with disease similarities. In this method, we used target gene-perturbed expression signatures as input data. Next, we prepared a predictive model for each disease. Each predictive model classifies input targets according to whether they are therapeutic targets or not. For trans-disease analysis, we learned predictive models jointly by sharing disease similarities. Finally, we predicted novel therapeutic targetability for diseases. We evaluated the performance of our proposed methods. We compare the performance between baseline method, inverse signature method, and trans-disease method. The left figure shows the result of inhibitory target prediction, and the right figure shows the result of activatory target prediction. You can see that trans-disease method performs the best for inhibitory and activatory target predictions. We performed target repositioning by using trans-disease method. Here is a network which represents the distribution of inhibitory targets repositioned from the original disease classes to other disease classes. Each node represents disease chapter in International Classification of Disease. In the inhibitory target prediction, many inhibitory targets were repositioned between neoplasms and immune diseases, and between neoplasms and musculoskeletal diseases. We examined the distribution of activatory targets. Many activatory targets were repositioned between neoplasms and endocrine, nutritional, or metabolic diseases, and between endocrine, nutritional, or metabolic diseases and musculoskeletal diseases. These results suggest that target repositioning can predict novel uses of existing therapeutic targets for different disease classes. This figure shows a small part of newly predicted therapeutic target disease association network. For example, MUC1 is an inhibitory target of multiple myeloma and breast cancer. MUC1 was predicted to lead therapeutic effects in multiple diseases, including atopic dermatitis, inflammatory bowel diseases, and systemic lupus erythematosus. We confirmed the validity of the therapeutic indications for 4 diseases with asterisks using independent resources such as recent papers and clinical reports. We also show the results of activity target prediction. For example, we focused on newly predicted associations of IL-10. Using independent resources, we confirmed the validity of prediction for 2 diseases, rheumatoid arthritis and type 1 diabetes mellitus. These results suggest that target repositioning can predict many promising therapeutic indications of existing therapeutic targets. I will summarize our work. We propose target repositioning to predict therapeutic indications of existing therapeutic targets. Target repositioning is expected to be effective in identifying novel therapeutic targets for a wide range of diseases. Thank you for your kind attention.